For this session. Firstly, before we begin, I would like to request everyone to switch off their cell phones and to all the presenters, please do not exceed your 8 minutes given time. Otherwise, we won't have time for questions. Now I'm going to hand over to our first presenter, which is Suzanne. Suzanne is going to talk to us about the ecology of sea urchin, a model for fisheries and ecological resilience.
Good afternoon, everyone, and thank you for listening to my presentation on my PhD project. So, hang on, sorry. Presentation notes on. Sorry.
Please present to you.
Will this still work the same? Just use my. All right, sorry, let's try again. Thank you so much for listening to my presentation on my PhD project on the Cape sea urchin Parachinus angulosus. Sea urchins in South Africa are an underexplored area in marine research, despite their ecological significance and potential economic value. Unlocking their potential hinges on a deeper understanding of their health, which is crucial for economic development and maintaining the ecological balance. By evaluating the marine ecosystem health, developing conservation strategies to protect vulnerable species, and finally, identifying potential stresses impacting sea urchin populations. To understand sea urchin health, I use the findings from three different approaches. First, a morphometric study, second, a genomic study, and third, a gut microbial analysis. I did it focusing on this urchin, which is the Cape sea urchin, Parachinus angulosus, as I think I have said twice now, a cosmopolitan South African species. On to aims and objectives. The morphometric study assessed the urchin's phenotype as a potential bioindicator of population health. The evolutionary genomic study used a genome-wide scan to reconstruct the evolutionary history of the species through time. Finally, the microbial community analysis, there we go, aimed to identify if specific gut microbial community structure can serve as an emerging bioindicator to assess the health of the South African coastline. So for this purpose, I collected 10 Cape sea urchin individuals from each of these 43 sites. Sorry, speaking to the mic. Starting at Port St. John's on the east coast and ending at McDougall's Bay on the west coast. So the morphometric study came up first, and it investigated the morphometric traits of the Cape urchin, measuring its shell dimensions like width and height, and that of the Aristotle's lantern, which is the mouth structure of the urchin. Though no dissimilarities were observed between coasts, some significant similarities were observed between populations within coasts. A separate data set focused solely on sea urchin spine measurements, measured and examined 150 of both the largest and smallest spines per urchin. The traditional methods of analysis did not reveal clusters at either population level or between the coasts. A Bayesian model testing a larger data set's accuracy in assigning sea urchin individuals to their population of origin was inconclusive, after which a supervised machine learning algorithm was applied using spine measurements. This had an average success rate of 66% in assigning individuals to their to their coast of origin. For example, when two individuals, one from Kids Beach and another from Coffee Bay, were used to test the model, both individuals were correctly assigned to the East Coast as their coast of origin. The second chapter took the form of an evolutionary genomics study. And here I extracted DNA from the intestinal tissues of the urchin, which was followed by library preparation using EasyRAD, a versatile method for restriction site-associated sequencing that is suitable for non-model organisms, which my species is. During the bioinformatic analysis, the enzyme used to cleave the urchin DNA also cleaved the mitochondrial DNA, which is, as the name implies, from the mitochondria, and is distinct and separate from that of the host organism, whose genetic information is found in the nucleus. A total of 25 complete mitogenomes were reassembled, which I used to reconstruct the evolutionary history of the species across the seascape. So here we can see that the origin of the species is in the vicinity of Cannon Rocks. And in the first wave, there was slight eastward and westward expansion of the species into the Natal and South coasts, respectively. More recently, a second wave saw colonization of the entire coastline into the distribution we know the species as today. It's likely that this colonization of the west coast happened during an El Nino event approximately 100,000 years ago, during which the temperature regimes on the east and west coasts were similar enough to allow for dispersed larvae to survive. Returning to the urchin genome study, the absence of clear population structure aligns with the commonly held belief that marine organisms, due to high gene flow and fewer barriers compared to terrestrial species, exhibit less evident population structure. Thus, analyzing genetic variation across the genome becomes the preferred method of detecting islands of divergence, seen here in the colored circles, which indicate significant variation between populations and potentially fine-scale adaptations. Chapter 3 investigated the gut microbial community composition using the 16SR RNA genetic analysis protocol, revealing 4 consistently present bacterial phyla: Fusobacterota, Campylobacterota, Bacteroidota, and Proteobacteria. Unfortunately, we did not find clusters between populations within each coast and definitely not between the coasts. Differential abundance analysis using balance regression highlighted one specific balance and explored the proportion of the bacterial taxa forming this balance. So, proportion of the numerator to the proportion of the denominator. The metagenomic functional content of these bacteria reveal their involvement in crucial digestive processes such as carbohydrate, amino acid, and energy metabolism. So what does this mean? Firstly, the morphometric analysis revealed variation between populations, but not between coasts. This aligns with the second chapter's findings, indicating that we have recent colonization of the West Coast, relatively recent, suggesting insufficient time for speciation between populations on the different coasts. The presence of the 4 core bacterial phyla in the gut support this notion, although it could also be due to the continuous water medium or the similar diets between the 2 coasts. My apologies. The South African Cape urchin lacks genetic differences, yet individuals from the east and west coasts face distinct current regimes. You have the warm Agulhas current on the east coast and the cold Benguela current on the west coast. So the west coast group may just be a transitory feature then, possibly facing extinction in the long run, as it is strictly speaking outside of their natural range. However, if we consider the effects of climate change on species distribution, might the water temperature on the east coast eventually exceed the upper thermal limit for this species, leaving the west coast a thermal refuge? We don't know, but thank you for listening to my presentation. I'd like to thank the following people and funders: And I'm now ready for questions.
Thank you, Suzanne, for your presentation. Are there any questions for Suzanne? Yes, Albert.
Thanks, Suzanne. Interesting presentation. In terms of the gut microbial communities, you said you didn't find any differences, but could that be related to the fact that your level of identification was much higher than, because you're looking at phyla, right? So if you could refine that to go to much lower levels, do you think you could possibly pick some differences?
Possibly, we have explored it and we haven't found such differences. For the purposes of condensing information into this presentation, I only included phyla, but we have explored that and it hasn't really reflected that.
Thank you.
You also have an online question, Sue. It's from Brian, it reads as follows. Q from Gavin Gauss. Nice talk, Sue. Having identified the islands of divergence across the genome, do you find any spatial patterns if you isolate those loci or any significant genome associations?
Okay, so maybe a little moment of truth. The graph that I showed, I did reference, it's not my graph. So we haven't actually explored that part of it. We've done preliminary analyses, but we didn't generate that particular graph. So I can't actually answer this question because we don't have that information yet.
Thanks a lot, Suzanne. You can go. Our next presenter is going to be Marcelo. Marcelo is going to present. His topic reads as follows: a first insight into the parasitic communities of Labiobarbus mariquensis within the Letaba River in South Africa, a baseline to determine the historical ecology of parasitism.
Good afternoon, ladies and gentlemen. This project is under the supervision of Dr. Marlies Trutter, Prof. Nico Smith, and Prof. Kerry Hatfield. Parasites are organisms that are very common but frequently unnoticed, unrecognized, and understudied. They play a crucial role in shaping ecosystems, and any changes observed in parasite populations may signify the dangers resulting from human interventions in a given system. So, how can we determine if a parasite community has responded to human impacts in a specific system, and when did these responses occur? This leads us to a new field of study known as the historical ecology of parasitism. This discipline examines nature over time, mainly focusing on human-environment interactions. With historical ecology, preserved samples could be mined for useful data, and the advantages of using these existing samples is that it reduces the need for additional sample collection and no field excursions are needed. It was hypothesized that there will be a change in parasite communities and diversity over time and community compositions of Labiobarbus mariquensis populations over time, and fish parasite diversity and the evenness of current fish populations communities will be significantly lower than in the 1960s. The following aims were determined for the study. Firstly, is to establish what parasite communities are expected from L. mariquensis by conducting a literature search of publications to see what parasites are known from this host in South Africa. Secondly, determining what river system could be used for the study. Secondly, is to determine present-day parasite diversity and community composition by sampling of fresh materials, complete parasite screening, morphologically and molecularly identifying taxa found, and determining diversity indices of species richness, abundance, and evenness. Thirdly, to study past parasite diversity and community compositions by screening museum-preserved fish samples for parasitological infections and morphologically identifying taxa found. Lastly is to determine the change in parasite diversity and abundance by comparing biodiversity from the past and present communities to each other and comparing the parasite indices from the past and present to each other. For fish collections, sampling has already took place at the Letaba River, which is within the Inkomati River system. The species of interest is Labiobarbarus mariquensis, a large-scale yellow fish from the family Cyprinidae. From museum collections, SAIAB has ample representatives from this river available in their collection, which potentially has a great number of parasites preserved within their fish hosts, which are previously not reported. These will be utilized to obtain a historical record of the parasitic communities within the river system. These two collections will enable us to know whether we should buckle up for more parasite outbreaks or if we are actually losing the parasite, or not actually, if we are losing the parasite diversity. Oh, sorry, yes. Elmeriquenses from the Letaba River were caught using cast netting and electrofishing. Individuals were humanely killed using blind force trauma followed by severance of the spinal cord, and there was an internal and external screening of parasites. According to literature, Elmeriquensis is known to be parasitized by monogenians, digenians, nematodes, crustaceans, trematodes, protists, and cestodes. 17 Elmeriquenses were collected, and the known parasites found during the present-day study include monogenia, nematoda, and digenia. Additionally, bivalve larvae and mixozoans were recorded for the first time parasitizing Elmeriquensis. Unfortunately, we could not find protists on the study. The following parasites were found. Firstly is the mixozoan plasmodia on the gills, and mixozoan spores existed from the gills. Secondly is the monogenia, also found on the gills, and here we have a representative of the whole specimen, the male copulatory organ, hamuli, ventral bar, and its marginal hooks. Thirdly is the nematoda found in the rhabdochona and nematoda found in the intestines, and we have representatives of the male's anterior and posterior end and the female's vulva. 3 digenian species were found, and with a highest prevalence of 59% and infection intensity ranging between 1 to 27. The first digenian was found in the cranial cavity and body cavity, the Telodelphus metasacaria, and we have its whole specimen and excretory pore. Second, found in the intestines is the Neodiplostoma metasacaria, and we have the representatives of its ventral sucker, oral sucker, and holdfast organ. The last one was found with clear cysts on the fins, the echinostoma. We have its whole specimen and secumoral hooks. Oh, lastly, we have the copopoda found on the gills, and we have its ventral view and dorsal view. Overall, the parasite prevalence was 65%, and infection intensity ranged between 1 and 27 with any parasite group. This parasitological study represents the first of its kind to be conducted at the Letaba River in South Africa and in Africa as a whole. This work provides novel geographical records of these parasites in South Africa, as well as new records of mixozoans and laval stages of bivalves parasitizing Elmeriquensis. Since the first insight into the parasitological study has already happened at the Letaba River, next will be the collection of preserved fish samples at the SAIAB Museum. Parasites found at the museum will be compared with the parasites that we found during the fish collections, and this will be able to know whether there was a change in the diversity and community compositions of parasites parasitizing Elmeriquensis. This research contributes to our understanding of complex interactions between humans and the natural environment, emphasizing the importance of protecting and conserving our ecosystems. Thank you. I would like to acknowledge the following. Thank you.
Thank you, Masilo, for your presentation. Are there any questions for her from the room? Let's take question A from the back.
Hello. Thank you for the interesting talk. I'm just curious about the specimens, the museum specimens that you're going to be looking at. So in terms of collecting specimens from preserved museum specimens, do you, when you do you dissect them, and I'm just curious as to what kind of state you leave the specimens in after having looked at them.
Okay, unfortunately, we will do a damage control and just dissect, because we need to look at all internal organs and also external organs for parasites, just so we can be able to know if whether they are the same as the ones that we are finding these days.
We still have time
Okay.
for maybe 2 more questions.
Can I
Um,
can I take a bite? Okay. I'm I've got 2 questions actually. The first is that you hypothesize that there's going to be historical differences before and now. Can you elaborate what informs that hypothesis? Because I mean, you said that you expect that diversity of parasite would decrease in the contemporary time compared to the historical time.
Yes, it's just, before you start a study, you do have you come up with ideas that, okay, maybe this might be the conclusion, or maybe this might not be the conclusion. But according to us, we are thinking that there might be a change, and maybe the parasites that were found before are not found now because of human activities that have been happening, because we'll be looking into samples from the 1960s. So from the 1960s until today, there has been a lot of anthropogenic activities happening throughout. So we don't know if whether those might have influenced the community or the diversity of the parasites that we have today. So we think that they might have influenced the parasites. That's why we hypothesized that there might be a change, and it might happen that the parasites that we found today are lower than the ones that were there before, because there were no maybe they were there, but they're not as much as they are today.
Okay. Then my second question relates to using museum collection and in terms of potential loss of information.
In terms of?
In terms of potential loss of information from those preserved specimens, are you concerned, or are you confident that what was the parasite infection rate then is still maintained in those collections?
Okay, I cannot say that I am 100% confident in that it will happen, but I have hope that we might find some parasites, even though there might be some losses that might have occurred. We don't know that. And this study has already taken place. I forgot in what country, in America. Yes, in America. And they did find some parasites in preserved samples. So we're hoping that in our collection in SAIAB, we will be able to find these parasites in preserved samples.
Thank you, Masilo. We have now run out of time. Our next presenter is gonna be Jabulani. Jabulani, his topic reads as follows: potential use of the indigenous knowledge systems for nature-based ecological engineering applications along an urban coastline, the early biofilm beginning. Thank you. Oh,
Good afternoon.
it's
I'm
recorded
representing a short-term
online.
study that forms part of my PhD. So natural coastal ecosystems are essential due to the wide range of ecosystem services they provide. However, anthropogenic activities such as urbanization, driven by economic development, is rapidly increasing in this region, bringing about various forms of changes, such as a replacement of natural coastal habitats with less complex habitats such as jetties and seawalls. And this is known to affect the initial colonization and recruitment of coastal species. So these changes are not only limited to structural changes, but also changes in water quality, where anthropogenic effluents containing high levels of metals and nutrients are introduced. This combined effect ultimately leads to the intensification on coastal species, lowering their ability to adapt and survive in these regions. Therefore, there's an urgent need in order to rehabilitate and maintain our coastal ecosystems. So in order to minimize or mitigate this effect, ecologically engineered solutions are there, which incorporate engineering and ecological principles to lessen these effects. So previous studies have mainly used cement in order to create eco-engineered structures such as plot senior and various types of cement tiles, and have mainly found an increase in species diversity and functionality. However, there still exists a knowledge gap on the use of a nature-based material in the creation of these structures. So as a first step on using a nature-based material, the study evaluated Cyperus textilis material and its indigenous knowledge application as a potential candidate for nature-based intervention. Therefore, the study aimed to create 20 by 5 centimeter plant structures by drying, sectioning, and combining Cyperus textilis stems in order to characterize that information and succession on the plant material, and also to assess metal accumulation potential of the plant material and its biofilm over time. So the plant Cyperus textilis was collected in the Hamburg district using indigenous knowledge system from the local community. And a total of 21 20 by 5 centimeter structures were deployed and collected at several time intervals to characterize diatom succession and metal accumulation. So at each time interval, environmental conditions such as physiochemical parameters, dissolved metals, nutrients, and chlorophyll A were measured in triplicate, as well as the biofilm was collected for diatom identification and metal analysis, and the plant material was also collected for metal analysis. And all statistical analysis were conducted using RStudio, where diatom, community descriptors, and environmental conditions were tested using either parametric or non-parametric tests. And for the metal analysis, a generalized least squares model was used in order to assess the relationship and potential effect between the plant and its biofilm over time. So we found no significant differences amongst the time intervals for physiochemical parameters, dissolved metals, chlorophyll A, indicating that the environment was fairly stable throughout the study. However, significant differences were observed in the levels of ammonium, silicate, phosphate, and manganese, and this could be either through natural processes known as sediment agitation, where nutrients or metals settling on the sediment are brought about into the water column, especially in turbulent environments such as coastal regions. Or this could be attributed to anthropogenic influences upstream of the Kawi estuary. So we found some interesting and beautiful diatoms, such as penny diatoms, including Chromatophora undulata, Navicula, Rhabdonema, Acnanthus, and a variety of Coconiensis species. We also found some centric diatoms, such as Actinocyclus, an unknown species, Paralea, and other diatoms such as Dalphia, Conconiensis, Lignophora, Pleurosigma, and Camplionensis. So with most of the diatoms identified are indicative of the species that have been previously reported present in the Kawi estuary. So during sampling, we observed algae growth after 1 month of exposure of the structures and potential grazing of a juvenile gastropod, which could be an oxystyle species. This observation could indicate that the plant material enable early stage species succession. For the biodiversity indexes, we found a total of 26 diatom genera, with significant differences observed only in species richness and also with genera diversity index. And this indicates that the plant material could house a variety of species, of diatom species over time. For metal accumulation, the plant material significantly accumulated aluminium and arsenic, whereas the biofilm accumulated all metals over the duration of the study. Interestingly, in the levels of manganese, manganese was depleted in the plant material, possibly indicating that the biofilm growing on it was using the plant material as an interface of accumulation, where accumulation could have occurred in either two possibilities by the plant and the biofilm, either through a process known as selective bioaccumulation, which is a more active way of accumulating metals, where essential metals such as aluminium, zinc, iron, and manganese were actively accumulated by the biofilm for biofilm's successive growth at different time intervals. Whereas another process could have been through bioabsorption, which is a more passive way of accumulating metals, where metals such as arsenic, which is a toxic metal, could have been absorbed by the plant through binding on the plant's surface or by the biofilm on the biofilm's extra polymeric substance. And this could indicate that the plant material and the biofilm growing on it could be potential bioremediators of arsenic. In conclusion, the plant material provided a suitable functional substrate as it enabled diatom formation and succession and also bioaccumulation of metals. So moving forward from the study, upscaling of the experimental design has been conducted by including additional treatments such as a simple structure observed there, and a complex nature-based design by seeding of muscles, and also assessing on a larger spatial temporal range in order to understand if there's an effect on time in terms of long-term exposure, and what happens when the structures are exposed to various anthropogenic influences in terms of site. So I'd like to acknowledge the following institutions and organizations for making this research possible. These are my references. Thank you.
Thank you, Jabulani, for a nice presentation. Are there any questions for Jabulani? There's one, there's another, but I think we can only take one question because of time.
I'll be quick. So you measured bioaccumulation of different heavy metals, and you found that plants did not necessarily accumulate. What is, yes?
Yes, the plant did accumulate arsenic specifically, and also which was a essential metal.
I'm just concerned about what you actually measured, because if you're comparing biofilm with the actual plants, surely there will be different roles in the where the material would potentially accumulate.
Yeah.
Could that be some bias in terms of your sampling?
In terms of, not sure if I'm understanding your question. So in terms of accumulation, yes, there's differences between the biofilm and the plant. With the biofilm specifically accumulating metals, different types of metals, depending on its function and where its stage in terms of growth is at. So the biofilm would generally accumulate either by the organisms within the biofilm, bacteria, algae, diatoms, or accumulate on the extracellular polymeric substance. Whereas with the plant, it could also be compartmentalized in different parts, either through binding on the plant surfaces or within the plant itself.
Thanks, Jabulani. Our next speaker is Pantoni. And this topic reads as follows: habitat use and movement of 3 sympatric mulet species revealed by acoustic telemetry. Over to you, Pantoni.
Thank you for the introduction, and good afternoon to you all. As introduced, I'm going to be talking about the habitat use patterns and the movements of mallet species using acoustic telemetry. So just a bit of a background. So also the species which are mallets, this is a group of species that are found in almost all the environments that we find in aquatic systems, from freshwater, the estuaries, and also in the marine systems. And they have various economic uses, from food, subsistence, recreation, and they also support some small-scale fisheries. And what you'll find with the majority of those mallets, the Mugilid family, is that they occupy transitional systems such as estuaries. And we all know that these systems are threatened due to activities such as overexploitation, from overfishing and habitat loss and degradation. And what you'll find in literature is over the years is that since these mallets, they support large biomass in most of the estuary systems that we find them, they have been identified as key ecological and environmental indicators for studies such as climate change. So if we look at the mallet species that we're going to be talking about in this presentation, so we find that their distribution around the South African coastline is strongly linked to temperature changes. So we find that like the southern mallet is in high abundance in the cold temperate region, and then also the groove mallet, you find it in abundance in the warm temperate region. And because of this, the climate change, which also affects the distribution of these animals. So it is important to understand the way these animals move within these systems to better understand how climate change may impact these fishes. And also, because of the multi-species approach that we're taking, it's also an opportunity for us to study how ecological concepts such as niche partitioning, as we shall see for these three species that we're working with, and also understand the drivers behind those partitioning. So there's three species that we're targeting. It's the striped mallet, the groove mallet, and the southern mallet. They are all endemic to Southern Africa. They are mostly smaller sized fishes, and they spawn between winter up to summer. And except for the striped mallet, which kind of grows up to 750 millimeters in total length. So the broader aim of this study was to look at the movement ecology of these three mallet species, just to understand how they move within the estuaries and how they are connected to other habitats, the freshwater environment, the estuaries, and also the sea. So the objectives is to determine the habitat use patterns and the connectivity of these species. And we're going to look at also how environmental parameters such as temperature will affect the movement of these species, as we're trying to link with how climate change might affect the movements and behavior of these species. And another thing that we're going to do, which we'll not talk about in this presentation, is to determine the critical thermal tolerance ranges of these species and try to link with the observed movement patterns. So our studies site is in the Kawi estuary, which is in Port Alfred. So we're using acoustic telemetry to track the movement of these species. And in the Kawi, we have set 25 acoustic receivers that are divided a kilometer apart from each other. And we've divided the estuary into three regions: the lower, the medium, and the upper estuary. And we also have temperature, salinity, and DO loggers in each of the regions of the estuary. So upon collection, we also did some genetic barcoding to determine which mallet species we actually caught, because these species are kind of similar morphologically, so we needed to make sure we caught the right species. So we managed to take 13 striped mallets, and fortunately we retained 13. And of the 22 grooved mallet that we tagged, we only retained 12. So that means the other 10, we actually misidentified them. And surprisingly, we also caught 6 southern mallet, which were not part of the initial study, and an additional we also caught a southern mallet, a blue spot mallet. And coming to the data, as you can see from the graph here, we have detections. So we've had many detections so far since we started tagging in February. And you can see the variation in the detections of each species and between individuals. You can see a lot of variation between the detections. And our preliminary results, what you can show is that from February up until October. So if you can see from the y-axis, as you move up, that's the section of the estuary. So you're going from the lower estuary going up. So what we've seen with these three species that the striped mallet in blue usually use mostly the lower parts of the estuary, and the groove mallet uses between the middle to the upper reaches, whereas the southern mallet, it's mostly restricted from the middle to the upper reaches. And then on an individual, if we look at the individual variation, so this graph is for the striped mallet. And as you can see, that's the pattern that we've found for the detection plot that we've found, that they are mostly using the lower parts of the estuary, although there are a few individuals that are also moving up the middle and also to the top half of the estuary. And with the groove mallet, we also see that most of them, they are restricted to the middle reaches of the estuary, with a few individuals going up and down the estuary as well. And lastly, the southern mallet, which we've seen that it prefers mostly the upper reaches of the estuary, and with a little bit, few individuals also going down. So what you've seen that with this data is that there's a clear partitioning between the habitat between these species. And so what we're going forward is to look at the drivers between what's driving those partitioning, one species preferring the lower, the middle, and the upper reaches. And one of the things that we're looking at, since we have salinity loggers in the estuary, is salinity. So the groove mallet and also the southern mallet, they have a wide range of salinity tolerance. So that could be the reason why we could find in most parts of the estuary, but the striped mallet is limited to the lower parts of the estuary. And other studies have also confirmed that they use mostly that lower part of the estuary, and they do not have a wide range of salinity tolerance. So that could be the case for that. And because of time, thank you.
Any questions for Bantoni? Okay. Okay, you can ask. There's a question.
Thanks, great talk. In a situation that one of your mullet got eaten by a big cob or a leofus, would you be able to distinguish that individual that had been taken out of the population? Because that tag would probably move through the alimentary canal of that predator in about 7 days and then be deposited on the estuary bed. So would you be able to pick that up?
Yeah, so I think with one of the presentation also there was somebody was highlighting that when you see that a tag is positioned on like the same location for an extended period of time, so you check the behavior, like what it was doing before, and then if you see that it's not moving for an extended period of time, you can assume that it has been eaten or it's been captured or something like that. Yeah.
We have an online question. I don't want to take 2 questions on the ground and then I leave behind the people online. Should I take the online? Okay, let's take the online question. It's also from Ryan. It's also from Ryan. Chantel Elston, thanks, Bantoni, great talk. Do you think the partitioning could also be related to differences in prey preferences?
Okay, thanks, Chantel, for the question. Yeah, there's a possibility that it could be also prey preferences. But what we've seen from the literature is that most of these mallets, they feed on a similar type of prey. But it could also be an opportunity for us to look at their stomach content analysis to just check whether there are differences in that as well, just to see the differences. Yeah.
Maybe we can also quickly take the question from Albert.
Thanks, Bandari. This is just a curious question from a freshwater taxonomist. So do they move between estuaries, or they are resident to the Kawi? I'm just curious. It seems the movements are just restricted or confined to the Kawi. Thanks.
Okay, thanks so much for that. Yeah, so what we've seen with the data now is that they are mostly resident in the estuary. So we're hoping that some of them can be picked up by the ATEP array, which is another network of receivers around the coastline. But we have seen with one of the, which we didn't intend on tagging, the one blue spot mallet that we tagged, it also went to Bushman's estuary. So there's a possibility that they can also move out of the estuary and go to another estuary as well. Thank you.
Thank you, Antonio. The next presenter is Tadiwa. Tadiwa is going to talk about the review of the Southern African genus Heteromomiras with the description of 6 new species. Over to you.
All right, good afternoon, everyone. I'm going to be speaking about the review of Heteromomiras within Southern Africa. I think a lot of you people have already seen this talk before. But anyway, onto this talk. So first of all, what is the importance of taxonomy? Like most of our biological studies that all of us are doing right now are underpinned by the knowledge of knowing that whatever you're working on, you know what species it is. So this can be within our own studies as well as in applied studies. You need to know exactly what you're working with. So that is why taxonomy is a very important aspect of what we do at SAIAB. So the next issue that we have, unfortunately, is that we have a lot of species in Africa, I have a lot of species that are not yet described. But unfortunately, the taxonomic capacity is something that has been, over the years, been declining because of various reasons, historical reasons. Also, not a lot of funds being driven towards taxonomy, fewer taxonomists being trained. So really, it's a case of where you have a big problem that's getting more difficult because there are fewer people to deal with the problem. So, however, it's not all doom and gloom, though. There are some people working on it. We are some of those people working on it. And we are actually trying to build some capacity towards resolving some of these issues. And yeah, also the improvement in technology, things like molecular techniques, as well as other techniques, imaging, CT scans, and all these other things are now tools that are in our toolbox that we can actually use to address more questions. Even though we are few of us, we can tackle harder questions that previously were very difficult to deal with. So for my study, I also was trying to tackle these three questions, very difficult questions. So the one that I started working on is the Heteromomiras. Initially, when I started working on it, it was actually in a different genus. It was Hippopotamus at the time. Then the other one is Brysanus lateralis, as well as Chiloglanus pneumoniae. All of these 3 species have a common feature that they are widely distributed and have disjunct distributions. And by that, I mean that you can see that there's these isolated pockets in their distribution. And from looking at that distribution, and knowing that fish have this physiological limitation where they cannot cross land normally in normal circumstances, the assumption was that maybe they may be different things. So I'm just going to talk about Heteromomiras for today. Well, yeah. So within Southern Africa, there are 3 valid species, namely Longilateralis, Zaboi, as well as Posiridatus. And then there are an additional 3 that are found within the Congo, one also Pappenheimi, and as well as Taveni. So when I started working on this group, I looked at Hippopotamus, it was Hippopotamus ansoji at the time, which has this peculiar distribution. And also in addition to the complication was the fact that we didn't know the type locality of this species. So the story of hippopotamus actually started with Kramer in 1996 when they did a study in the Upper Zambezi and they looked at the species. And a cool thing about momarids is that they produce weak electric organ discharges, which are very weak electric currents that they use for exploring their environment. And it's a character that has been like explored a lot because, I mean, a fish that produces electricity, who doesn't want it part of that? So that was one of the first things they did. So they collected allozyme data from the Upper Zambezi and they discovered what was possibly a new species. Then they followed it up with the study and they actually looked at the EODs and they found that they found two different distinct patterns. And from that, they were able to describe Hippopotamus zaboi, this one. Following to that, Kramer and Schwartz in 2010 did another study in the Kunene River and they found a new species as well, Hippopotamus longilateralis on the Kunene River. Then more recently, Dr. Albert did a study from the Eastern Zimbabwean highlands looking at the Pungwe, Ruo, and Ebusi River system. And from there, they were just looking at a genetic barcoding study. They were able to identify new lineages from the Pungwe, the Buzi, as well as the Ruo here, including the other ones that were already known, like the longilateralis, as well as some from the Kwanza River. So then they recruited me into doing the masters, and I then had to look at the entire thing because they just looked at a small part, and I looked at the entire range of Heteromomaris. And at that point, from my study, we actually found there were at least 9 unique things that were genetically distinct at the time. So yeah, this is how they're distributed. And as you can see, most of them, there are 5 of them found in the Kwanza River, whilst the rest of them is individual groups found in individual river systems, which was very exciting for me because I was like, yay, 12 new species to describe, but was always more complicated than that. So at the time, that was Hippopotamus. A more recent study by Sullivan et al., they looked at a specimen from the Kwanza River. So in the Kwanza, there was one which was called Heteromomaris postradiatus. It was known from just one specimen. And we always wonder, like, if these things are found across the entire Southern Africa, how can we just find one specimen in this river? Where are the others? So they looked at then they looked at the skeleton. Then they realized, actually, first of all, they had fancy techniques. They were able to actually extract DNA from a museum specimen, which was pretty cool. Then from there, they looked at the skeleton and they found out that actually it was deformed. That's why we couldn't find others. So essentially, we could then link it to some of the lineages that I had found, which is K4. So now going into my aim of the study was to now just build onto all this information and actually get to the point where we love to get as a taxonomy, which is the actual description of the new species. So of course we had to do our integrative taxonomy, integrating genetic work, nuclear genes, CO1, as well as the morphology. And from my study, we then were actually able to describe 6 new species, of which, well, yeah, paper is still on its way, but we did find some distinct characters, including scale counts, vertebrae counts, as well as various differences in proportions. And particularly if you look at the shapes of the heads, they actually are quite distinct for some of the groups. So that's where we are at now with this group. So with these 6 species, they're all, we're able to actually assign posterior data based on Sullivan study. Then we're also able to deal with Heteromomaris and Soji, which was the big problem to begin with. Now we know where it's found, which is also the Kwanza River, as well as there's a resident one that's there, which is Papanemi, as well as 2 new guys that I'm describing. Then we have one from the Kunene, Okavango, the Upper Zambezi, Pungwe, Buzi, Ruo, all have individual species. So in short, taxonomy is very fun and you get to describe new species. And in Southern Africa, we now have an additional 6 new species. And yeah, thank you very much for watching.
Any questions for Tadiwa? Any questions online? Oh, there's a question.
Thanks. It might be a simple question, but would you think that your 6 species originated from a common ancestor? And if so, was that original species much more widely distributed? And then these individual species have speciated in those river systems over time. Is that the logic?
Okay, at the moment, we haven't looked at that specifically, but that's where we want to get, because right now we're just assembling basically the alphabet. Then after that, we can actually make sentences and tell the story. So, okay, like I said, we haven't looked at that specifically, but just from the top of my mind, I assume that's one possibility, because within Southern Africa, the rivers have actually changed a lot over time due to geological events, erosion, rivers connect and change. So what the rivers look like now is not what they were like 20,000 years ago. So it might have been a widely spread species that eventually speciated as the rivers actually broke up. Another possibility is because of their unique ability to produce electric organ discharges. Other studies from other groups, like for example, Campylomyrmex, have shown that they also can speciate in sympatry, because of this unique ability to actually communicate, just like the birds. They are actually, you do find some sympathetic speciation also occurring in some groups. So they are actually very interesting patterns that we can probably find out from this. But right now, within Southern Africa, we're still just trying to find out who's who and where are they found, so that we can actually answer those very interesting questions.
We've ran out of time now. Maybe you can ask him the question during the comfort break period. The next presenter is Siphelele. Siphelele's topic is comparing natural and artificial coastal habitats, insight from complexity, soundscape, and physiology.
All right. Afternoon, ladies and gentlemen. Thank you so much. Let me get straight into it. So in the background, coastal environments are very productive systems, and ecosystems such as rocky shores, I need to get used to this mic. Ecosystems such as rocky shores are known for their intricate structures, which, when influenced by various biotic and abiotic factors, and that fosters abundant biodiversity. Due to this high complexity of interactions, the dynamics of such complex systems can vary at multiple scales and be observable at the patch level too, which then is shown by the various pictures that I've put on the corners. And then ultimately, this overall complex configuration helps maintain populations of marine benthic organisms in coastal ecosystems. Now, the importance of coastal habitats is they provide structural and surface complexity in the sense of provisioning or providing a variety of habitats. And that gives rise to nursery and spawning grounds, microhabitats, which can be critical for early life stages of animals, in this, or in my case, invertebrates, and then this increases biodiversity. The other importance is that they also provide unique sound signatures, and the sound in general is important because it helps with communication, it helps with orientation to suitable habitats, describes the quality of the environment, and also it helps in detection of prey and predators. And then I'll finally end it off with thermal refuge, as some marine organisms, or several marine organisms, are ectotherms and therefore are dependent on the environment for survival and development through their early life stages. So the rationale behind this is that coastal urbanization is increasing. That's one. This leads to expansion of coastal infrastructures and therefore a rise in replacement of natural habitats. Now, these coastal infrastructures could be jetties, breakwaters, gabions, such the like. And coastal infrastructure does give rise to artificial habitat. However, artificial habitats are not exactly the same as natural habitats in the sense of the kind of materials they're made of, physical, chemical properties, microbiome, and also ecology. So the aim of my study was essentially to compare natural and urbanized coastal systems to determine the quality of these environments for invertebrate larval settlement. So specifically just for this talk, I'll be determining or speaking on the structural complexity. Then there's also the characterizing of soundscapes and also assessing the fitness, which is the physiology for early life stages of invertebrates and fish. So these are my study sites, two natural sites, which is Cannon Rocks and Kinney Bay. We have the two big harbors, Port of Mocha and Port of PE, and then the two smaller harbors or two smaller ports, which is Port Alfred Marina and Port St. Francis. So with the complexity, I want to measure the three-dimensional complexities of the rock surfaces that and Cannon Rocks. In most or many of the scans that I was able to make with this machine, I found that on the scans themselves, there's a majority or a whole lot of mussels, barnacles, and all sorts of other invertebrates that are on the substratum itself. And that's both true for Kinney Bay and in Cannon Rocks. And obviously there's very different curvatures and should we wait till it comes back? Okay. And then we have, then there's artificial sites, which is Port Alfred Marina and Port St. Francis. They are kind of similar. Thank you. Now I can point. So Port Alfred Marina and Port St. Francis, they are kind of similar in that they are particularly made up of boulders, because the artificial sites, they really don't care about the biodiversity. It's a matter of protecting jetties or people's properties and all of that. So it's more boulder-like. That's for Port Alfred Marina, and that's for Port St. Francis. And then with the last two, which is Port of PE and Mocha, surprisingly for Port of PE, or where we are working, is that these little, I'll call them mini hills in the sense, these are actually mussels, a variety of mussels and barnacles, and sometimes oysters. And in here with Port of Mocha, though it is a ditch, you do get some sort of settlement of a lot of variety of invertebrates on these rocks. Just to show you then some preliminary results, I did a sort of one-way ANOVA with the site versus the surface area, because I did come across some issues in trying to actually analyze some of the scans, but this is what I'm going to talk about. So there was a significant difference across all the sites in terms of the surface area. And just to show you on the graph, so the two green are Cannon Rocks and Kinney Bay, so those are the naturals. Then we have the two reds, those are the two big harbors, PE, Port of PE and Port of Mocha. And then we have the orange, which is St. Francis and Port Alfred Marina. Yeah, as you can see, these smaller ports, especially with with Port Alfred, is that it provides a larger surface area, which may be a potential available substratum for animals or for the inverts to come and actually recruit on, as compared to the other sites. Now, a bigger surface area does mean that there's more potential available space for settlement and recruitment. However, that does not necessarily mean there's more biodiversity. And what I can allude to is that there are other several environmental factors that actually play into this. Could be water quality, can be temperature, which we are currently monitoring. It could be available biota such as the mussels, which are considered as bioengineers that can further shape, and potentially even the other anthropogenic impacts such as sound and other things as well. But when you look at Port Alfred and you look at St. Francis, like I said, the boulders, they're much bigger and they're much more clear. So when you scan, it does seem like there's more space, but it doesn't necessarily mean there's complexity within the rocks themselves, because there are certain taxa that are habitat specific. You know, you don't find limpets in the entire intertidal zone. They are very specific. Barnacles can be very specific where they choose to settle, and that also includes mussels. So I think it needs much more different points to look at, such as surface elevation, rugosity, and all of that. But as I said, I've had issues with the scans that I've made previously. So I'm thinking perhaps I need to use a different software in terms of analyzing these scans, and potentially redo the scans in the sites, which is not a real train smash, because it was kind of like a once-off sampling. So I just need to do that. But other than that, I want to acknowledge all these organizations that have assisted us, and these are just my references. Thank you. And thank you, load shedding.
Just one question for Siphelele.
Siphelele, the use of quadrats sampling, you said you randomly throw them. How random is your random sampling? Is it random, or it's strategic random?
I think it's, to be honest, I think it's strategically random, because even as I was speaking, I said I threw them, but I actually threw them within the space that they've set up, the nature-based structures that they have attached. The whole entire project is within the intertidal zone, but specifically where the mussels, the native and the invasive co-occur, so almost in the mid-region. So within that region where they've set up even the structures, that's where I've thrown the quadrat. So it's not truly random. There is some form of structure to it.
Yeah. So because I was going to say, perhaps it's time to talk about other sampling approaches, like stratified random sampling, and things like that.
Okay. Thank you for that note.
Thank you, Siphelele. The next presenter is Jody. Jody is going to talk to us about the first insights into the population genomics of the critically endangered 74 sea bream. Over to you, Jody.
Thank you. Okay. Alrighty. Okay. Hi, everybody. So as we all know, South Africa has a highly dynamic marine environment, and a rich marine biodiversity. And in particular, the south and east coast of South Africa, with its 5 known barriers to connectivity, is quite a suitable region to study population connectivity. And this is essential to determine whether our current MPAs, marine protected areas, are effective and for further spatial planning purposes. So as we all know, our ecosystems are protected by 41 MPAs. However, to date, they still lack the inclusion of genomic data. And several of these MPAs were even implemented to protect several important fishery resources, such as sea breams. However, a study done by Hendricks et al. In 2020 actually found that the current network does not protect the full evolutionary and functional potential of spadix. So as you can see in the figure, Basically, the blue represents a spatial overlap of species richness, endemism, phylogenetic and functional diversity, and the green is the current MPAs. And as you can see on the south coast, there is no overlap between MPAs and those indices. However, the use of genomic data with its high resolution, one is actually able to investigate inter and intra-population structure. And the last MBA even noted the need for novel biomonitoring methods of threatened marine taxa, such as sea breams, and with the 74 sea bream being a perfect example. As many of you know, this is a South African endemic, and due to fishing pressures, the stocks collapsed in the 1960s, with a moratorium being placed on the capture of the species, and it's still currently in place. And it is rated as critically endangered on the IUCN Red List. However, there has been recent evidence that noted that the stocks may have recovered, and basically highlights the need for novel biomonitoring methods to assist in the reassessment of the species. So this brings me to the aim of this component of my PhD, which is to investigate the population connectivity of this Polystegonus angulosus along the south and east coast of South Africa with the following objectives: to use genomic data to assess the population structure, to assess demographic trends, and then also incorporate this data into the spatial planning and management of priority taxa such as sea breams. However, for the purpose of this presentation, I'll only be focusing on objective 1. So for the methods, so currently we have 19 whole genome sequence data. So 3 from Durban, 11 from Kai, and 5 from East London. We're still awaiting sequence data, 10 sequence data from Kai and 1 from East London. However, yes, there is a typo. It's not garden route, it's garden route. We would like some additional samples in the blue squares. So if any of you have some fin clips that was ethically collected, please do speak to me after this. Yeah, so I just recently got my data. So for the purpose of this presentation, I only analyzed the whole mitochondrial genome. So, and I refer to it as mitogenome. So basically DNA was extracted, library was prepped using whole genome sequencing at a 12.5 coverage, sequenced on Illumina HiSeq. The sequences was assembled de novo, and then it was annotated and aligned in Mega, and we did various analyses. So prelim results, one cool thing, and which was unplanned, was that because the sequence data was so good, we were actually able to assemble and annotate the complete mitochondrial genome of Polystegonus angulosus. And this is a first for the entire genus. And as you can see that it is roughly 16,600 base pairs in length. It has 13 protein coding genes depicted by the color green. Yeah, there's green, yellow, like a light pinkish, and purple. One thing that people need to take a note of is the famous CO1 gene. Okay, anyways, is thank you. Yes, the barcoding region, the CO1 gene, that is only 9.4% of this entire genome, meaning that my whole genome sequence data is actually 28 times more informative than just looking at that one gene. So the genetic diversity indices, I'll just go through this quickly. The neutrality test gave negative but significant stats. AMOVA, of course, would give a higher variation within the sites. I don't know why this presentation looks like this. Anyways, so basically what you're looking at is a haplotype network. Basically, figure A is just the same representation of the haplotype network in figure B. So basically, because this is whole genome sequence data, Each mitogenome is unique, so depicted by 19 samples. So we have 19 unique haplotypes. The little small lines, I know you can't see it, just means one little hatch means one mutation. So even though, I mean, at this large scale, it's surprising that there's really small differences between them. Also, the PCOA and the Phi pairwise also showed some difference between Kai and East London. However, we're looking at a small data set, so there's no clear trends as of yet. So in summary, even though we use a small subset of our data set, 19 whole genome, whole mitochondrial genomes is still able to pick up differences. The negative neutrality stats basically showed a similar pattern to a PhD study done by Oosthuizen. However, he just looked at the ND2 gene, and this result may be interpreted as population expansion. And though each individual's mitogenome is unique, they still picked up sequences, small sequences between them. Of course, the use of genomic data will help improve the knowledge, fisheries assessment and management of the species. Going forward, of course, we're going to try and increase the data set, and that will help depict clearer trends. And then the use of the whole genome sequence data, the nuclear genome sequence data, we'll be able to investigate how inbred the species actually is, and also to assess any demographic trends such as the effects of fishing pressures. And yeah, I would just like to thank the following people and organizations. Thank you.
Any questions for Jody?
Thanks, Jody. Great talk. I was just curious, are you, so two considerations, I think. I'm not sure how easy it'll be to get 74 from the west of Algoa Bay, but you'll very easily be able to get them in Algoa Bay, and Koos will probably be able to get them for you. So that's an easy place to add samples. Okay. I don't know if you're thinking about that already.
I would like some, yes.
Yeah, so that'll be an easier win for you because he's there as well.
Okay.
So you should speak to him about that.
Okay.
And then, are you engaging with Sven Kuwith and fisheries?
Yes. So they are planning on doing a sampling trip. I just don't know when. But yeah, he's still busy planning. Hasn't.
Okay. Great. Yeah. Thanks.
Thank you.
Thanks, Jody. Nice to see you. Yeah, thanks for this. It's all new to me, so it's exciting to see this. I was just interested to tell that story that we found with the decline of the population over the 20th century. The work that Carl did in his PhD thesis, showing that the sort of heterozygosity of 74 had dropped from the sort of end of the heyday of 74 fishing in the 1960s to 2006, 2007, 10 years after the moratorium, implying that the population, effective population size had decreased a lot. And I'm hoping now that with your study and the collection of these samples, we'll be able to now compare to what we found in 2005, 2006 with what's happening now. Is that still on the cards for you? Are you still going to be able to look at that?
We would like to, depending on resources, financial resources, sequence, whole genome sequencing is quite expensive. But yeah, we would definitely like to. I am still in contact with Carl. So yeah, we would like to explore it in the new year. Okay, that's it. Thank you.
One more question.
One more.
I'm lying. It's from Ryan. Nice talk, Jody. Taking the whole genome component further, is the intention to use PSMC approaches to get at the demographic history, or is it more traditional SNP-based pop genetics? And then what platforms and approaches are you using to get the genomes?
Okay, I'll try and answer. So yeah, we definitely do want to explore, demographics. So basically any effects like fishing pressures on population genomics. So we are looking at SNPs, so not just the traditional population genomics. Yeah, so we are trying to explore what actually happened with the 74 over the course of years. Yeah, then a pipeline. So I guys, I was still doing analysis in the week. So I haven't explored pipelines as of yet, but that is the next step, is to look at the nuclear genome and analyze it. Yeah, thank you.
Thanks, Jody. The next talk is online by Angus. It reads as follows: understanding the interoperability and complexity of disparate fish visual sensors techniques and optimizing data value for stakeholders.
Good morning, everybody. My name is Angus van Wyk, and today I'm going to be presenting a chapter from my ongoing thesis titled Understanding the Interoperability and Complementarity of Disparate Fish Visual Sensors Techniques and Optimizing Data Value for Stakeholders. So in hindsight, I probably could have simplified this and sort of titled it something like an honest attempt to help monitor our fish within marine protected areas when we reach our targeted 30 by 30. So just as a quick introduction, there are several things that marine protected areas benefit from. So one of them being size of the reserve, the age or maturity of the park, its location, for example, protected area's proximity to an established fishing community or one that has a history of non-compliance, and of course, clear boundaries. So unlike their terrestrial counterparts, marine reserves have no fences. So having mechanisms in place to educate the public and the commercial sector about the whereabouts of your boundaries are key. But ultimately, what marine protected areas benefit from is a positive relationship between management processes and ecological impacts. So that's good data in to a capable management structure, which can then be used to inform all interested stakeholders. So from your interested South African citizens to state policymakers. And getting this process right and overcoming these challenges is key to the success of marine protected areas. And this is essentially what I'm tackling in my PhD. How do we make better use of the tools available for marine protected monitoring in order to provide a more comprehensive data set to managers? And how can we overcome some of the key challenges facing management by building a stronger network, ultimately increasing the data value for all relevant stakeholders? So what are some of the tools that we use to monitor fish within our marine protected areas? Well, many of you would have already come across a baited remote underwater video system, perhaps from our great talks earlier today. So these are our primary video monitoring tool used in South Africa, but we also use diver operated video or underwater visual sensors, such as diver fish counts or snorkel surveys, and made famous by our local coelacanth population, remotely operated vehicles. So how do we make better use of these tools? Well, we need to ensure that these tools are interoperable or can be used together, and put structures in place and ensure that the data collected is complementary. And this is important because what we're seeing is that diver operated video and underwater visual sensors have proven highly effective at quantifying cryptic and small reef fish, which is a relative weakness in BRUV's datasets. But a strength of BRUV's monitoring complements UVC by including large predatory species and elasmobranchs, or your sharks and rays, into a dataset. And of course, including data at depth that is not safe for divers. And ROVs, which have proven effective in monitoring deep marine canyons within our marine protected areas. And something that struck me last week is how relevant this is as we aim for the 30 by 30 target. This is going to push research and management into new and potentially unfavorable marine areas. Many will probably be very challenging to monitor, and policymakers are going to need all the data that we can get them. So that's a brief introduction to how we can make better use of our tools. But what I want to go into a bit more detail today is our first steps towards assisting the management process by building a stronger visual sensors network. And our first step towards building this strong network was to gain an understanding of our community within the network. So who exactly are we? How do we communicate? Are we on the same page and speaking the same language? And what discussions are we having? Are there topics that we find incredibly important? And are we not focusing on discussions that we potentially should be? So to better understand the community, we decided to conduct an epistemic analysis using a free-to-use tool called Cortext. And just as a note, I'm really just going to glance over the method. So please, if anyone has any more questions, feel free to reach out after the talks via email. The analysis begins like a standard systematic review with the construction of search terms and the collection of bibliographic data. We included texts from almost 1,500 online resources dating back to the early 1950s. We then use this data to create a large text corpus and use natural language processing to extract the most important terms used by the community. And then once we prepared the bibliographic database, built the text corpus, and extracted the terms, we were able to make use of the scripts within Cortex Manager. So like the fun begins. And the first being geocoding, which is able to assign geographic information to raw author affiliation addresses found in your citations. And that allowed us to answer our first question. So who are we as a community? Well, it turns out that we're a fairly large and interconnected collection of 88 different countries. We shook hands and agreed upon topics over 11,000 times since the 1950s. And our community leaders, in inverted commas, appear to be Australia, the United States of America, and the United Kingdom. But we're not a static community, but quite dynamic. And like many scientific fields, we're also prone to shifts in geographic biases. So that's a snapshot of who we are, but we also wanted to know if we're speaking the same language as a community. And as it turns out, we're 15 official languages, or more formally known as knowledge clusters. So ranging from seagrasses to mesophotic depths, from sharks, rays, and skates to herbivorous species. But our primary languages are marine reserves and reef ecosystems. So although the only cluster which may be described as isolated, those researching rock pool ecosystems, it's important to point out the insular characteristics of the reef ecosystems cluster. It has the largest nodes within the network, but not many connections. So there's a significant amount of research going on within this cluster and a lot of discourse, but they're beginning not to speak the same language as everyone else or the rest of the network. And I'm sure many of us could sort of see themes like this within our different fields. And effectively what they're doing is distancing themselves from the community as a whole as a result. So that's our community and those are our languages. But as it turns out, we also have dialects. So each of the visual sensors methods has a sort of unique dialect or its own discourse, with browsers characterized by terms such as no-take areas and management, diver operated video by reef condition, UVC by coral reefs and reef fish, and ROVs by mesophotic reefs and offshore oil. So that's how we communicate in a nutshell. But what other contemporary conversations are we having? So this is an epoch analysis displaying our most important terms and how they track back through time. So just at a glance, one can notice how many more discussions we're having as a community sort of in the last 10 years compared to the 50 that came before it. And our most important discussions seem to be focused around reefs. We also have a strong focus on management and marine protected areas, on sharks, fisheries. So although fisheries is discussed more often, it has seen a decrease in its importance, which is quite interesting. And fish communities as a whole and not single species, which is a key monitoring strength of visual sensors. So those are important discussions, but we also wanted to know how the larger discourses have evolved through time, and have we stopped having or muted once important discussions? Well, it turns out that management and marine protected areas are one of our longest running discussions. So these are important to us as a community, but so too are coral reef ecosystems and the commercial sector represented by the gas and pipeline industry. What's interesting in this analysis is the evidence to suggest that deep water reef research is stepping out of the coral reef shadow and carving its own path. Perhaps this is made possible by advancing deep water technologies, or perhaps this is entirely driven by Anthony and Kerry Sink's eagerness to explore deeper and deeper. Who knows? But as it turns out, that once important discussions within the network are heavily muted, such as fish behavior being discussed in the 1990s. But what we're also not seeing yet is the established discourse involving environmental DNA. There was an impressive amount of sort of online chatter about eDNA and its promise to bolster monitoring using visual sensors, but this seems to be muted currently. And so too is citizen science. There is great potential to involve citizens in marine monitoring using visual sensors, but we're not having enough of these conversations to have a loud enough voice within the community. So that's us. What's next? So in an attempt to put structures in place to strengthen the network, we are using the information from this chapter to host a number of workshops and send out an international questionnaire to the global community. So it's fairly obvious that we are only going to be able to monitor our fish species over a much larger spatial scale if we work together as a well-functioning community within a network. So please, if anyone is interested in attending, please reach out.
We can take, thank you, Angus, for the presentation. We can take one question for him. Any questions online? Okay, Angus, I have a question for you. On your first few slides, you showed us some graphs. What impact does visibility have on the use of graphs, and what do you do to overcome them?
Okay. Yeah, so that's obviously one of the, I guess, weaknesses, as you could say. One of the hamperings about using underwater visual sensors is, of course, visibility. So, I mean, what is unusable visibility? So say getting a zero sample, you can't use it at all, is actually pretty dark. So say around about 1.5 meters viz, as we would sort of call it quits on a video. So that's not bad viz. And how we overcome it is, it's a lot of the times we put it in our models. So visibility, we have a 3D measurement, it's gauged by distance that we can see an object. And yeah, when you put that as a factor in your analysis, yeah, visibility will play a massive role on your abundance measures or your biomass. So that's, yeah, for the most part, how we overcome it.
The next speaker is supposed to be Melissa, but her presentation is cancelled, but Francesca has an announcement to make.
So we were supposed to have a comfort break, but I think I see you comfortably enough sitting there and we're running out of time. So just a couple of announcements. Please return the name tags. It's plastic. We want to use them again. As you exit, there's going to be a box that you can collect and you can return them to the registration desk. Can you also return the Plickers paper so that we can use them again? And what's going to happen now, there's still 2 presentations to go, but I would like to give you some instructions for voting with a proviso that you're not voting yet. So we need to get through these 2 last presentations, but there's going to be 2 so there's going to be different ways in which you can get your vote. Whoever is attached to SAIAB will receive links onto the SAIAB WhatsApp group. Whoever doesn't, they can come here and scan a QR code on the phone, one in red for supervisor. Supervisor means postdoc and above, from pensioners, whoever. And the blue one is for the students. So if you're not having any access to our email or online or WhatsApp group, please come here quickly and scan your QR, and then please don't vote yet because we still have 2 presentations to go. And so we're only gonna accept votes as the end of the last presentation is completed, and then we're gonna move on to our last keynote speaker. Everything clear? So if you need just to scan, come over here. Otherwise, I think we should be receiving things online as well. Thank you. I think I'm gonna send it because I haven't received any. Do you have it? I know, I don't want to be, I want the. Because I had it before. I used to have it and I deleted it.
The next presenter is Martinez. His topic reads as follows: revalidation of Enteromius crocodilensis from synonymy with Enteromius argenteus, with some preliminary insights on the biogeography of Southern African large soul-fin barbs. Over to you, Martinez.
Thank you for that. And thanks for sticking around so late. So Tadiwa spoke nicely about the importance of taxonomy and what we do. So I'm not going to go into much of that. And this might not be as exciting as describing a new species. And I've talked about our new species enough, I think. So this is a small part of my PhD where we show that Enteromius crocodilensis is a valid species and separated from Enteromius argenteus. So the Enteromius are the genus of small diploid barbs endemic to Africa. You can identify them, tell them apart from some of the other cyprinids by their scales being radially striated, and they've got 7 to 8 branched dorsal fin rays. And there's a lot of taxonomic issues in Enteromius. It's a very large genus, morphologically quite similar, very few identification keys, problems accessing sampling locations, and then same with Tadiwa's work, many species have widely or disjunct distributions. And so my story here begins with Enteromius argenteus that was described by Albert Gunther in 1868, based on specimens collected by Friedrich Welwitsch that you might know from the Welwitscher plant. And these were described from the Kwanza River system in Angola. And then in 1934, this Henry Weed Fowler described a species collected from the Sabi River in the Nkomati River system. Not the Sabi, the Crocodile River, hence the name. We did this description based on a single specimen. And back in those days, prior to genetics and everything, I think some people didn't like the idea of specimens being described just on one specimen, the species being described. And so the problems came that Enteromius argenteus is never mentioned in the literature in the early 20th century that occurs in South Africa. And it's the first mention is by a chap called Groenewald from the Leidenburg Fisheries Institute. Couldn't find a picture of him. Who in his revision of the barbs, as it was known, as Enteromius was known back then, he said the taxonomy of the Barbus group was confused by Fowler's ill-defined species. And in this revision is the first time argenteus is mentioned in the literature as occurring in South Africa. And he doesn't talk about crocodilensis other than in the same diagnoses as with argenteus. And from there, Rex Jubb on the left there, sent some specimens from the Crocodile River up to Humphrey Greenwood in at the British Museum in London to have a look at them. And Humphrey Greenwood apparently looked at them and said they're the same species. And so Rex Chubb synonymized it, but there's no evidence that we actually looked at it, I think. We just take his word for it. And so when they synonymized Crocodilensis with Argentius, we just got this disjunct distribution between the Kwanza and then South Africa, the Nkomati system in Pongola, where this Crocodilensis occurs. And again, for freshwater fish, such a distribution is quite strange. And then Albert threw the project at me. We did some genetics. It's not very clear there, but Crocodilensis is in yellow, Argentius is there in red. And from the genetics, you can see it's clearly two different species. Did some measurements, and based on the morphological measurements, there's quite a lot of overlap. Argentius is in black there, Crocodilensis in red. And based on the morphology, there were some patterns that we could see separate the two species. Argentius has a higher dorsal fin height than Crocodilensis. Argentius has a deeper body compared to Crocodilensis. And then there's some other characters that you can pick out. The head shape's different. The dorsal fin, I can't see it more there, is in Argentius is a lot more concave at the top. And then if I can get this pointer to work, on the lateral line scales, there's these chevron-shaped markings as well in the Crocodilensis specimens, which kind of leads me to think that I don't think Humphrey Greenwood and Rex Chubb looked at the right specimens or even looked at them properly. And then meristically, with the meristic counts, they separate out very nicely. And these are based on the circumpeduncular scales. In Crocodilensis, 12 circumpeduncular scales, and Argentius 14, and the same with predorsal scales, 11 to 12 versus 9 to 10. So definitely we can pull out Crocodilensis as a separate entity, which then allows us to redo the IUC and red listings to look at the conservation of these groups, and then quickly talk about the biogeography of this large shorefin clay that we've picked out of my Enteromius phylogeny. The Argentius is up there, Crocodilensis, then you get Matosi in the Limpopo system, and in the Kunene, Manicensis up here, Coloensis, and Enteromius lutamba. And it's got this weird distribution that's all around the upper and middle Zambezi and the Okavango, where you don't find any of these species. And my thinking is that the Kunene used to flow down into the Etosha and was later captured by a coastal stream. And the Etosha was linked to the Paleo Lake Makgadikgadi, which was a mega lake which persisted up till about 35,000 years ago. And I think maybe there was a linkage, obviously, through the Okavango, and with climate fluctuations, the Okavango area and the Kalahari dried up and went extinct in this area. Anyway, thank you.
It's time for questions. Are there any questions for Martinez? Are there any questions online? Okay, I do have a question. You said that Enteromius argentinus has a disjunct distribution. What could be the cause of that? And or rivers sampled for you to like make those conclusions? Is it enough sampling from the literature?
So the Argentius itself doesn't, it used to, well, when we called it both populations, when we called both populations Argentius as the same thing, that gave it this disjunct distribution. So now by revalidating the name Enteromius crocodilensis, it doesn't have that distribution anymore. So Argentius is we restricted to the Kwanzaa system, and crocodilensis is restricted to the Nkomati, Pongola, and Mkuzi systems.
Thank you, Martinez. Thanks. The next presenter is Nonhle. Nonhle's topic reads as follows: developing a framework for predicting the future distributions of coastal fishery species. Over to you, Nonhle.
Hi, everyone. My name is Nonhle Tubelihle Mloja, and my study, or rather, I'm presenting my PhD proposal, and the title of my research is Developing a Framework for the Prediction of Future Distribution of Coastal Fisheries Species. And these are my supervisors. So climate change is defined as long-term trends or shifts in temperature and weather patterns that have an impact on the marine environment. Now, these long-term trends are thought to be a result of short-term thermal variability, such as drastic changes in temperature, prolonged cold spells, and heat waves. Temperature is considered to be the greatest stressor in marine organisms, as these changes have a major impact on the distribution, demographics, and behavior of marine fishes. The overexploitation of coastal fish stocks in South Africa's marine shore-based fisheries is placing the sustainability and health of our marine environment at risk, as exploitation exacerbates the effects of climate change on marine fishes, and it is thought to remove larger, older, and highly thickened individuals from fish populations. The synergistic effects of exploitation and climate change have a negative impact on species recruitment dynamics, leading to age-truncated populations. Now, the extent of how the current and future climate variability will affect the distribution of coastal species is unknown. This is due to the lack of information on the spatial distribution and the potential impact of climate change on our marine shore-based fisheries, which is important to know the current and future distribution of marine fishes against various climate change scenarios. And this can be done using species distribution models. What are species distribution models? It is when we seek to understand how and why biological organisms are distributed in space by quantifying the correlation between environmental variables and the distribution of organisms. We have two types of species distribution models. It is your correlative SDMs and your mechanistic SDMs. Correlative SDMs assume that the current distribution of a species gives a good indicator of the ecological requirements of that species. It uses a statistical relationship between long-term summarized environmental temperature and observed species localities. Then your mechanistic SDMs incorporates fine-scale biological processes experienced by fishes to assess their response and behavior, and it uses detailed physiological data from species obtained through laboratory simulations of current and probable environmental conditions. So types of data that are required for these models. Your correlative SDMs use presence and absence data, which is data obtained from ORI and RESPO. They use long-term occurrence data, spatial data, and environmental data as well. And mechanistic SDMs uses output physiological data from experiments, ecological processes, and species biology. Now, over the past 10 years, the Rock and Surf Super Pro League, RESPO, in partnership with the Department of Ichthyology and Fisheries Science, has managed to record data collected from competitive recreational anglers in order to inform improvements in coastal conservation and management strategies. And the Oceanographic Research Institute Cooperative Fishing Tagging Project has been tracking the movement and growth rate of fish through tagging since 1984 to date. This is done in order to implement wise management and sustainable use of our marine lionfish species and resources. So the data from RESPO and ORI is what we're going to use to inform our mechanistics and our correlative SDMs. The bronze beam is endemic to Southern Africa and occurs in shallow rocky coastal areas. It is one of the dominant species in recreational and small-scale fisheries of the southern coast. The catch rate of this species for the past 30 years have substantially declined. The species has therefore been declared by the IUCN as near threatened. It is therefore our study species. The aim of the study is to develop a framework to guide the use of correlative and mechanistic species distribution models that map the current and future geographic distribution and biological characteristics of our overexploited marine shore-based fisheries species under predicted climate change scenarios. And to do this, we will carry out physiological experiments such as a critical thermal tolerance experiment, which will look at the critical thermal limits of fish, upper and lower critical thermal limits of fish. We'll then do metabolic rate and critical oxygen saturation limits experiments using respirometry. Thereafter, the results from those physiological experiments will be used. We'll use the physiological indices identified from those lab experiments to develop mechanistic species distribution models. We will then run correlative species distribution models to inform the development of a framework in predicting the current and future geographic distribution and biological characteristics of our marine shore-based fisheries. We will then compare the mechanistic SDMs and the correlative SDMs in order to predict the movement of fish with changes in temperature. Our study site, fish will be collected along the Eastern Cape, warm temperate coast starting from the Great Fish River all the way to Cannon Rocks. So our materials and methods for the critical thermal tolerance limit, we will assess the thermal, we will have to identify the maximum and minimum thermal tolerance of fish by subjecting them to temperatures that are outside their thermal range until the fish lose equilibrium. Then aerobic scope. The fish, we will have to identify the standard metabolic rate of fish when they are resting, and then the fish will be chased. After that, they will be put back into the respirometers, and then we will measure their maximum metabolic rate. To get the aerobic scope, we will subtract the standard metabolic rate from the maximum metabolic rate. And thereafter, we will measure the critical oxygen limit of fish by cutting off the water supply to the respirometers and measure the rate of decline until the fish lose equilibrium. Our correlative SDMs, the model will be run using the Datamo high performance cluster provided by IFREMA in France. We will then identify bioclimatic variables that are important in defining species distribution. Then we'll map out the known distribution using the occurrence data and CPUE data obtained from RESPO and ORI. So this is the CPUE data that we got from RESPO. So it has been recorded from, it has been recorded from 2013 to 2003. So the brighter shades indicate the higher CPUE occurring within the areas. So this competition takes place in the Western Cape and the Eastern Cape from False Bay to the Wild Coast. That is why we don't have anything recorded in KZN, because it is here. And this is, am I here? Okay, it's fine. Yes. Okay, so the potential outcomes of the study will help us focus the distribution pattern of marine species and their chances of survival when faced with thermal variability. We'll also provide knowledge of the thermal limits of South African lionfish by comparing the physiology and species distribution of an important species, which is Eupatrimetopon grande. This will have a major impact in economic and social significance and alleviate poverty in communities reliant on these fisheries for income as a source of protein. These are the research activities that I'll be doing throughout this project. These are my acknowledgements. Thank you to everyone, and thank you for your time and attention.
We can have one question for Nonhle.
Thank you. That's an interesting project that you have. One of the predictions for climate change is not just a gradual increase in temperatures and so on, but an increase in the frequency of extreme events such as marine heat waves. Are your models capable of incorporating extreme events?
I think so, because we are going to get data from SAIAB that has been recording temperature data, like sea surface temperature data, in the places that we will be sampling. So there we'll be able to identify your thermal extremes, cold spells, and whatever drastic changes in temperature that we might find. And then we'll use the sea surface temperature data, I think from Copernicus, and try to compare those. And also the mechanistic distribution, the physiological experiments that we'll be carrying out. We'll look at how fish respond to those low temperatures and then compare with the temperatures that are recorded by SAIAB.
Lovely. Thank you.
Thank you.
And now there's a question. Oh, okay. There's one more question, Nonhle. So
There's a question from online from Pule. Since your title is climate change, is there a specific reason why you're not including ocean acidification as well, or do you think it will be negligible?
We don't have a reason, but so far we've been looking at the effect of temperature. But if we might look at ocean acidification, if we just to see how fish will respond to it. So we're not opposed to including it. We might not. So thank you for the suggestion, Pule. Okay, thank you.
thanks again, Nonhle. I'm now going to hand over to Francesca to introduce our keynote speaker.
And there we go. Don't we say Dulce Fundo for the sweetest for the last? So I'm just going to give you a little information about Lyle. Quite proud. Sometimes I feel still like a proud mama about Lyle. Dr. Lyle Forsett is currently a marine biology lecturer at the Department of Biological Sciences at UCT. He finished his PhD at Rhodes University while he was based at SAIAB, where he investigated the impact of mangrove microhabitats on the larval ecology of decapod crustaceans and fish, as well as explored some aspects of their ecophysiology. Following this, he pursued postdoctoral studies at the University of Hong Kong. Remember, guys, it's important to go out and explore. Remember what Christopher said, leading to a project addressing macro and micro plastic pollution in critical peri-urban mangrove ecosystem. His current interdisciplinary research focuses on the ecological consequences of human-induced pollution, habitat degradation, and climate change. He explores how these factors shape the community structure and ecophysiology of both adults and larval invertebrate fish and fish population, contributed to a holistic understanding of multi-scale ecosystem dynamics. So this is our last keynote. So let's just give it a round of applause for Lyle.
Well, firstly, I want to say thanks for sticking around, and thank you for that introduction. It's an incredible honor and a privilege to be giving this talk to you guys today. And I'm really impressed by the quality of talks that has been happening today in South Africa as well as on the continent. Unfortunately, I'll be taking you guys to East Asia today. We will board a bullet train to go through the anthropogenic pollution in East Asia's mangroves close to the northern limits. Okay, so we are currently living in the Anthropocene. It is the era marked by human activities shaping the Earth's ecosystems through industrialization as well as urbanization. And the evidence of this impact on our ecosystems is unmistakable. We can see it in our coral reefs bleaching, we can see it in the land cover that we're losing of our rainforests, and we can see it in fishing stocks being depleted. These coastal habitats, or coastal habitats in general, aren't immune to these stresses as well. These stresses such as overexploitation as well as habitat degradation have negative consequences for coastal ecosystems, but today I'll just be zooming in on anthropogenic marine debris, talking about metal pollutions, and indirectly talking about climate extremes as well. So plastic has become an integral part of our lives, right? Due to its low cost and its versatility. Over 9 billion tons have been produced in the last 70 years. And because of its ease of use and its low cost, it's resulted in us having a single-use lifestyle. I'm a hypocrite because I'm standing here with a plastic bottle of water. There are generally 3 ways which we deal with this plastic waste accumulation. One is we recycle it, or we think we recycle it. Number 2, we incinerate it, which can have harmful effects to our health. And 60% of all plastics produced end up as disposal, right? So how does this plastic that you dispose in our environments? Well, there's 2 ways. One is inadequate waste management systems. These are insecure landfills, and these are illegal dumping, which results in environmental leakage. The second way is irresponsible and inadequate waste disposal, right? And after storm events, you have runoff and river discharges, which takes these plastics or this this debris to our coastal ecosystems. And we've all seen pictures like this before, right? As soon as it enters our ecosystems, it interacts with fauna, and the consequences are usually negative. So globally, our climate has been increasing cyclically. So there's a trend of increasing temperatures, and it culminated this year in the Northern Hemisphere having the hottest summer on record. So we anticipate this heating to continue. And what we do know about organisms is that they operate in a limited band of temperatures that they can deal with. Right? But besides this effect of heating and the stress that it puts on organisms, there's also industrialization and urbanization, which results in metals pollution as well. If we look at global water bodies and how polluted they are, and just to give you an idea, in China, there are water bodies that have pollution figures in terms of iron and manganese higher than the water drinking standards from the World Health Organization. Closer to home in South Africa, we also see that we have a very, very high proportion of chromium and nickel in our water, also above World Health standards. So if it's harmful to us, this means it must be harmful to organisms as well. And these metals have a very, very negative effect in that they disrupt our biological processes. So these are just the 3 anthropogenic stresses I'll be talking about today. So what are mangroves? Well, mangroves are these wonderful trees that are woody or shrubs within the intertidal zone. And they usually have 2 general belts. One is towards the surface, or towards the seaward side, which basically faces the sea, the bay, or the estuarine mouth, which it's situated in. And number two is the landward side, which is faced towards the terrestrial habitats, or sometimes in the peri-urban habitat communities. So mangroves are critically important because they provide numerous goods and services. They provide food, they provide timber, they provide critical nursery habitat for ecologically and economically important fish and invertebrates. But they also help in regulating the Earth's climate, and they sequester about 5 times more carbon than terrestrial forests. So where are these mangroves distributed? Well, mangroves are distributed between 30 degrees north and 30 degrees south latitudes. The largest land cover of mangroves is in Southeast Asia, particularly in Indonesia. And then we go to the Americas in Brazil, and Australia follows closely. So these mangroves basically operate in a temperature band. So they thrive in the tropics and the subtropics and are limited by the 20 degrees Celsius average annual mean temperature. So at its most northern limits, this is where we're going, is Hong Kong. And then, of course, South Africa is here at the south, which is also one of the southernmost limits of mangrove distribution. So when we think about Hong Kong, we think about this urban, sprawling metropolis, right? It's a southern Chinese metropolis. It's a commercial center. And it houses 7.4 million people. To put this into context, it's one of the most population-dense places in the world. There are about 7,000 people per square kilometer. To make a comparison, South Africa only has 49 people per square kilometer. But Hong Kong is unique in the fact that it's not just this skyscraper skyline that you see. There are these pockets of green space, and particularly mangroves, and they have about 500 hectares of them, which encompass over 60 stands, of which Maipo is the largest. It is also a Ramsar wetland of international importance. But it is situated along the southern mainland Chinese border, as well as being influenced by the Pearl River Delta. So the Pearl River Delta is one of the top 10 polluting rivers in the world. So you can imagine what type of influences it's having on that system. Besides having so much land cover, they also house an incredible biodiversity. 58 crab species within 12 families, which are critical for ecosystem function in that they help in nutrient cycling, predator-prey relationships, as well as the overall ecosystem health as well. So we wanted to know, what is the extent of this urban population having on environmental marine debris leakage? So we looked at 8 mangroves, we looked at 5 on the east and then 3 on the west. And what we found was that there's a spatial variation in the amount of marine debris that's getting deposited in these mangroves. Well, firstly, the East Coast is getting more debris, larger debris, so which means that the surface area covered by debris in these mangroves are larger than the West. But the West Coast is getting more items, which means that the items are just becoming smaller and smaller. So we wanted to find out if this density of population is a driving factor of the pollution trends we're seeing. And it is not. So we overlaid population density, and we found that population density is actually a very bad predictor. So when looking at site levels, we wanted to find out if the 2 general belts have a significant difference in the amount of debris that's being deposited in them. And we can see from the graph that the landward zone of these mangroves are harboring much more debris than the seaward zones. Hong Kong has 2 distinctive seasons. One is the wet season, which is during the summer, and one is the dry season during the winter. So what we found was that seasonally, this abundance of marine debris is localized. So you have to go to the site. You have to look at the site. It's not a general pattern. And what we think is that the seasonal winds could be an influence in depositing debris in these individual mangroves, because Hong Kong is influenced by the East Asian monsoon, which brings very strong northerly winds coming from mainland China. Whereas in the wet season, you have very weak southeasterly winds. So this could be a factor. When looking at what is actually being deposited there, well, there's no surprise that a majority of it is plastic in terms of number of items and the surface area that's being covered. To dive deeper, we see that hard plastics or plastic injected mold plastic is dominant in the landward zone of most sites, as well as foam plastics such as your polystyrene. We also see that soft plastics such as plastic bags and wrappers dominate the seaward zone. We also tried to categorize and quantify exactly what the plastics were used for in the first place. And what we found was that in terms of surface area covered, the items that were used for domestic purposes were not the majority, but the largest single category. So this could be plastic bags, brushes, anything that you use for your domestic purposes, followed by unknown. The unknown category were items that were way too small to categorize confidently. This is followed by industrial uses such as metal that were clearly used in industry or processed wood that has been illegally dumped. So that was by surface area and then by number of items. It makes sense that the items that were smallest were also the majority in terms of number. But there is also a spatial variation in terms of region. The West is more dominated by domestic use, and the East, which is known for its aquaculture and fishing industries, is dominated by fishing debris and industrial debris as well, which we speculate is from illegal dumping. So we know what is found there, but at which rate are these debris items accumulating? We set up around 3 experimental quadrats and 3 control quadrats and basically monitored these for a year. From June 2021 to May 2022, going in every month, cleaning it up, counting it, identifying it, measuring it, weighing it. And then we found similar trends. We found similar trends to what our initial monitoring study told us. The landward zones is definitely a sink, and the landward zones is definitely accumulating more plastic than the seaward zones. We also noticed that there's a dry season peak in terms of how fast it's accumulating in these mangroves. So this just provides a little bit more, well, evidence in saying that the northerly winds brings most of the debris into the mangrove. So this is all good and well. We know what's there, we've characterized it, we've worked out its accumulation rates. But is it actually impacting the biodiversity of these mangroves? The short answer is no. So generally, we found that plastic had no effect on the biodiversity metrics that we worked out. I forgot to mention that we also monitored the fauna, the invertebrate fauna within those quadrats every single time we went. And we worked out biodiversity metrics such as Shannon's diversity metric, Simpson's diversity index, species richness, as well as below evenness index, which is the commonness or rarity of a species. But there is a couple of anomalies, but we don't know if this is an artifact, which showed that in terms of surface area being covered by debris actually results in a positive increase in biodiversity metrics at the landward zone in Tinkock. We think this is an anomaly, but there's of course a competing hypothesis, which states that as you increase the complexity, even if it is marine debris, you'll get more species trying to exploit those spaces. So this warrants further investigation. But it could also just be that the mangroves aren't that polluted when you look at the scale of pollution across the world. We've seen the negative effects of pollution having deleterious impacts on communities on beaches, as well as on mudflats in Ireland. So it could just be that we need to emphasize exactly where are these tipping points where even if it's an anomalous positive impact, does it become negative and then start to impact the ecosystem negatively. So we know there's macroplastics. So there's bound to be microplastics as well. And we set out to find out how much and what influences the loadings that we're getting. So in terms of in surface waters, we found no differences, no spatial differences, no site-specific differences, and no seasonal differences in terms of microplastic loadings in the surface water of these mangroves. It could just be that the microplastics coming in are just biofouled very quickly and then sink to the bottom. But what is concerning is that the microplastics in these surface waters are the highest in the region, for mangroves at least. What about mangroves? What about microplastics in the sediment? Well, again, we found no regional differences, but there were site-specific impacts. Notably, we have 2 sites that have very little microplastic loadings. It could be a product of the saying that you are a product of your environment. So what kind of activities are happening close to these mangroves? Well, the 2 that have very little microplastic loadings is Paktam Chung, which is a country park, so it's heavily policed and it's protected. And the other one, which is Tinkok, is also a bay, but it has a massive highway separating the closest village to the mangrove. We can see that everywhere else there is a landfall, there's fishing activity, there are clam digging, there's surfing, windsurfing, or they're barbecuing right in the vicinity of these mangroves. Again, spatially, there is a landward, there is a propensity for the microplastics to be in the landward side, which again confirms that landward zones of mangroves are sinks for macro and microplastics. And the wet season had higher microplastic loads than the dry season. And this could be because of after a rainfall event, there's higher surface runoff from communities surrounding the mangrove, and there's more urban river discharges as well. So we know the mangroves are there, but are the most important animals in the mangroves eating them? The answer is yes, but there is no species-specific differences in the amount of microplastic ingested. But there is a slight trend. We can see that crabs, which occur, Parasasama bidens, which occurs in the landward zone, has a slightly higher microplastic loading in its gut than the Trinita crenata, which occurs in the seaward zone. And this is a reflection on how much microplastics is in the environment. It could also be a reflection on the feeding behaviors. So Trinita crenata is a generalist, generally eats mollusks and other crabs. The 3 fiddler crabs are microphytobenthivores. So they basically eat microalgae on the sediment and they filter the sediment out and possibly filter the microplastics in that behavior. And then you get the Parasasama bidens, which basically just eats anything it sees, but mainly is a leaf shredder. So it's shredding leaves and eating detritus. This is confirmed in a study that my colleagues did in 2020, which also found a similar trend where Parasasama bidens had the most number of microplastics in their guts and their gills. So we know there are microplastics, we know there are macroplastics. The fauna are eating it, but they're also interacting with it within their feeding behaviors. So are these macrofauna contributing to the microplastic loads that we're getting from the macroplastic debris that's being deposited? The answer is yes. We set up an experiment to find out if 2 common ecosystems engineers, namely Parasasama bidens and Metapograpsis quadridentatus, are interacting with plastics, and if they basically fragmenting plastics to a point where they're contributing to the microplastics. So we gave them pristine blocks of polystyrene, as well as biofouled blocks of polystyrene, and saw how they got on over 7 days. What we found was there was a propensity for Parasasama bidens to interact more with the pristine blocks, and it fragmented more microplastics than with the biofouled blocks, as well as the Metapograpsis. The Metapograpsis, on the other hand, interacted more with the biofouled blocks. And shredded more plastic than the pristine blocks. We think that this could be a consequence of their feeding behavior, as well as the morphological differences in their claws. So Parasosama obediens is a leaf shredder, so it generally has a claw that has a cutting, so it's serrated. Whereas Metabographis has a spoon-shaped claw because it feeds on algae, it scrapes algae off the rocks. So this could be an indication of how it's interacting with these polystyrene blocks. So anthropogenic marine debris, microplastics is generally a visible threat. But what we forget about is the invisible threats as well in the mold of heavy metals or metals in general. And mangroves are predisposed to accumulating these metals because of their sediment binding capacity. And they therefore may act as a reservoir to transfer metals from the sediment to higher trophic, to producers and then to consumers. So we need multi-scale data. We need data of the sediment, we need data of the plants, and we need data of the consumers of the plants. And we set out to do this in 5 mangroves in situ. And what we found was that in Maipo, probably the most important mangrove in the region had the highest contamination of all mangroves. And this could be a reflection on the Pearl River's influence. It basically correlated with every single metal being increased. So this means that there are contamination hotspots, and we have to be cognizant of these spatial differences. So aluminium was the highest, and cadmium was the lowest as well, and cadmium could be toxic in very, very low concentrations. When looking at the uptake of the plants that are in these mangroves, or the population of plants that are in these mangroves, we can see that there are 1, 2, 3, 4, 5 species that we looked at, and manganese was the most abundant, regardless of species, and cadmium was the least abundant, regardless of species. What we also noticed was that there was no significant site differences. So this means there's an uncoupling from the local sediment to what the plant is actually taking up. So this is from fresh leaves. So there could be a step in the pathway where it actually excludes the metals from its leaves. But there are also species-specific accumulations, which probably speaks to the essential metals that particular species need and their physiology in excluding certain metals. When looking at the macrofauna, we see that the fiddler crabs or the ocypodids have the highest concentrations of metals generally, especially the toxic ones. I just also want to say that there was no difference in terms of site or across species, but there was a trend for families. We think that this is because of their burrowing behaviors. So ocypodids are crabs that generally maintain their burrows every tidal cycle. So they're interacting with the sediment a lot. But this doesn't explain why it's higher than the sesamids who also build burrows. So, possibly another situation where there are competing hypotheses. Right? So different families had different heavy metal loadings. So it's not equal the way it's proliferating up these trophic levels. Then we wanted to know if these metal loadings are impacting the populations of crabs negatively by looking at their physiologies. So basically we collected Parasosama obediens from these 5 sites. And what we found was that the population from Maipo had suppressed metabolic parameters. So they had lower Pmax, which is your maximum metabolic rate. They had a lower thermal optimum, and they also had a lower critical temperature. So what does this mean? Well, this means that if there is extreme events, the Maipo population, which is, of course, again, the most important mangrove in Hong Kong, main ecosystem engineer could be facing deleterious impacts. We also then, from those crabs, we took their muscle tissues to see exactly how concentrated the metals were in their muscles. And we took the muscle tissue from their claws to make it standardized. We tried to regress these metabolic parameters, or the oxygen consumption parameters, and we found that as cadmium increases, maximum metabolic rate, and mean oxygen consumption decreases. So cadmium is actively suppressing the metabolism of crabs at MIPO. But if you look at the table, you see that MIPO doesn't have, the crabs at MIPO don't have the highest concentrations of cadmium. So what's going on? Or any metal for that example, for instance, except for manganese. Well, we think if you look at the standard deviations over here for cadmium, so it's generally super low, 0.1. And here we have a standard deviation of 1.8. So we're thinking this could be an outlier, masking the effects or masking the averages for the different sites. We also found that cadmium and lead account for about 40% of the variation seen between the populations in the maximum metabolic rate, and only 23% of the mean oxygen consumption. The thermal optimums were not impacted by heavy metals as drivers. So what is the scale of pollution in one of the most densest populations in the world? Well, even though they're very dense, they have excellent waste management systems. And for this reason, the AMD pollution is relatively low compared to the rest of the world. The pollution is spatially driven at both the regional as well as the local scale. The primary source of anthropogenic marine debris is mismanaged waste, and the Pearl River is not such a great influence. Having seasonal trends can inform concerned citizen groups. I've never ever seen a poster that says mangrove cleanup at this date at this time. Only beach cleanups matter, right? So anyway, concerned citizens groups can have the trends of when debris is accumulating the most, and then make a plan to remedy the situation. What are the impacts? Well, it's the microplastics at the moderate level in both surface water and mangrove sediments, and the microplastics mirror the macroplastics. So it's a very good indicator of microplastics loading. The influence of the macroplastics degrading and fragmenting is likely the greatest contributor. And it has about moderate to high levels of contamination on the west, but luckily not on the east. But what we need to be cognizant of is that as coasts continue to develop, both plastics and heavy metal pollution may increase. And this even counts for Ramsar sites because this threat is invisible. So what are the impacts? Well, in Hong Kong, it's not having an impact on the biodiversity yet. We need to find out if there are density thresholds. But if the surface area is covered, this means there's less surface area for crabs and gastropods to burrow and to feed. So at some point, it's going to be negative. The crabs are ingesting microplastics, but they are also a source. We're also seeing that the microplastics may affect their individual energy budgets and behavior, and also make the microplastics available to smaller organisms. In terms of heavy metals, mangrove leaf metal concentration doesn't matter about what the sediment looks like. There is physiological responses of certain species that excludes heavy metals in its tissues. Mangrove crabs take up the heavy metals and may be a sentinel for environmental pollution and effective bioindicators for mangroves. And chronic exposure to non-essential heavy metals like cadmium may reduce species' thermal windows. And this is particularly concerning because we can see now that keystone species and vital ecosystem engineers are affected by these threats. And if there are extreme events, they may fail in their functional roles, as mangroves already have a very, 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 very low functional redundancy rate. I'd just like to thank my collaborators who pitched in for this effort, Prof. Konichi, Dr. Knott, Rebecca Butler, and all my swamp dwellers as well. Thank you so much. But over time.
Thank you. Thank you so much, Lyle. It's wrapped in, was that your postdoc?
Yeah.
Yeah. Sounds like another PhD. Any questions for Lyle? I've got one online, but let's take one from the floor. This one from Christopher. I've got.
Hi. Sorry, thank you for the great talk. I just have a question with regards to how you classified your plastics when you were separating from where they actually came from.
Yeah.
And then with your answer, I have a follow-up question,
Sure.
maybe even suggestion. Sorry? Okay. So yeah, how did you classify your plastics?
Sure. So we worked with a table. So crockery, pots, pans, brushes, anything that we deem as domestic. It's a very subjective and qualitative process. So anything that was pans, pots, crockery, etc., was domestic. Then we had industrial, which were basically metals and processed wood. So we know that this might have came from factories. Or it was in the industrial process, so it's not natural. Then we had unknown, which were debris that was so small that we couldn't confidently say, okay, this could be used as for something, right? So it's very too small, it's unknown. Then we also had fishing, so that would constitute fishing nets, cut, hooks, for example. We also had industrial fishing, domestic, unknown. I think that was the 5. Yeah, 4, 5. Yeah. Follow up. We have a publication that I can chat to you about, and you can look it up as
We
well.
can just talk after this.
Yeah.
You can follow up.
Yeah, you can follow up.
So with regards to classifying the plastics, have you ever thought for the ones that you cannot identify using FTIR and using them on a, identifying them on a polymer level? Because with that, you are able to construct risk assessments associated with those plastics for and the effects that they could potentially have on the organisms, because you don't know. You just sort of go. Do you understand what I'm saying?
Yeah. I mean, that's a great question. Where we find a stumbling block with that approach is that it's very time intensive and it only gives you the polymer. It doesn't really tell you anything about what it was used for. So for instance, a film wrapper that's packaging could be domestic, but it also could be packaging for something that's industrial, for example. For instance, bubble wrap. So bubble wrap is usually used in an industrial setting when you're sending stuff away. So we classify that as industrial instead of as domestic use. So it will be the same polymer as your plastic bag or your whatever you're using, you're eating your wrappers, your chips or whatever wrappers. Yeah, so that's kind of the stumbling block. Yeah.
While the microphone reaches Christopher, I think I'm going to take a question that was on the chat that I received it. Sorry, it's too far. Maybe the microphone is reaching Christopher. Okay, from Jabulani. Hi, Lyle. Thanks for your presentation. I wanted to know or find out if there was or if there is any correlation with previous or current studies between the type of microplastic you found with any other form of pollutants, such as metal, persistence, organic pollutants, between the sites that you were working in.
It was mainly different sites, right? The heavy metal pollution stuff was in different sites than the microplastics pollution. So it would be difficult to do a correlation, but I'm sure a heavily industrialized and a heavily urbanized area or ecosystem or mangrove or whatever ecosystem you're looking at will have a high number of microplastics. But there's also a very, I don't want to say a big probability, but there's a chance that there's heavy metals as well. As far as it's correlated with, there's a relationship in terms of they're there because one or the other is there. I mean, we can't say, but something to think about, I think, I guess.
Lyle, this is probably, excuse me, probably something that you haven't looked at because that was a massive project that you did. But there's another invisible threat which we've done some work on, and that's microplastic leachates. And they have a strong effect on the behavior of various animals like mussels, for example. Have you ever looked at anything like that?
I have not, and it's definitely in the pipeline. How are we going to get ethics clearance to do those experiments? We'll have to see. But what I do notice is in the literature, when you're doing an experiment, what people are using is non-additive microplastics, which means those leaches are absent. So they don't really get an effect of what could potentially be leaking from those microplastics, right? So it's something to think about, yes. But that's probably in the future.
If you are thinking about it, what we've been doing is contrasting the effects of virgin microplastic pellets with ones which have been beached and which have been accumulating pollutants while they're in the sediments and so on.
Okay, I'll follow up with you on that.
Sorry, I'm trying to be multitasking where we try to organize the prizes for the winners and things. And but if there are any further questions, we can, for Lyle, we can take them. Otherwise, we proceed with the final processing of the symposium. Any more questions? No. So I think I would like to invite Siphelele and Sue on stage. Siphelele. And while things are still getting organized, yeah. So the easy bits is that we want just to give a little present or sign of appreciation to our keynote speakers. So Christopher and Lyle, can you please come on stage to receive your little present?
Thank you. I thought this was my present. Does anybody want a microphone?
You can't take it home. You can't take it home. That's not your present. So next category. Thank you so much, guys. Okay, now it becomes difficult. Lucky's gone. He's abandoned me. Don't leave me now. So I think what we're going to be doing, we don't have runner-up. We are on cost containment. Sorry, guys. So what we will do, we're going to have a first prize for the best MSc presentation that the student voted and the best MSc presentation that the supervisor, postdocs, that broad category voted for. So there should be something being projected from my side. And yes, so basically Chandra wins. Actually, she's got a double thing because she won unanimously the prize from students and from a supervisor for the best MSc presentation. Come on stage, Chandra, please. This certificate that you see on the screen, you will receive it imprinted. No, sorry. It's fine. I don't know, are they the same? Yeah. So just give one. No, because we've got a surprise and we're running out of prizes here. So that's. Come, Sue. Yes, come. There we go. I feel like at the Oscars. So this is like MSc category. I'm not quite sure what's happening with PhD because I was listening to live, answering questions. And so I think I'm just going to go with whatever certificate comes behind my back. So we got Vuyo. Oh, hold on. There is a surprise. This is the best presentation according to who? Where is lucky students or supervisors? You come on stage, you're getting something.
Please tell me.
Supervisor, but wasn't there like a surprise on that? Sorry? Wasn't there a tie? So can we have the, and Nanche Motwa is also coming on stage because she's getting the best PhD presentation from supervisors. So come on stage. We like this box. Yeah, well done. Yeah. No, we got a final student. I hope there's only a winner from the students because we've only got one prize. So surprise, surprise, who's winning the prize for the best PhD presentation voted by the students? No Bushle Mpanza. Okay, come on stage, no Bushle. So that's it. You're not going yet because we like being official. So I'd like to one last time call Albert on stage to just give the very, very final remarks. And I just want to give my remarks after we set up a little bit here. I'm just going to head to the red for a drink. So everyone is welcome to join and continue the celebrations. Thank you very much.
Hi again, everyone. I don't really want to be the one standing between you and that nice ice cold beer at the Ritz. So yeah, just a take-home message. I'll just get this from what Chris said. Good science is the hallmark of civilization. We had some really fantastic presentations here. Just want to urge you all to continue with the good science that you're doing. Thank you for hosting such a successful event, Francesca and your team. Thanks, everyone. Yeah, that's it.